Good afternoon. My name is Mingyu Zhang, and I'm a PhD candidate from Johns Hopkins. The topic of my presentation today is in utero exposure to metal mixtures and offspring blood pressure, and analysis of the Boston birth cohort using Bayesian kernel machine regression. There are no conflicts of interest to disclose. It is no secret to my audience today that high blood pressure is the leading modifiable risk factor for cardiovascular diseases. Blood pressure tracks from childhood to adulthood, and higher child blood pressure is associated with cardiovascular diseases in adulthood. So this really reminds us that targeting environmental risk factors for child high blood pressure may serve as a primordial prevention method to prevent cardiovascular diseases in adults. Lead, cadmium, and mercury are metals that cause adverse health effects at trace levels. Exposure to these metals is associated with higher risk of hypertension and cardiovascular diseases in adults. On the other hand, manganese and selenium are essential antioxidative elements for human health and are suggested to have protective effects against cardiovascular diseases. These metals and elements can cross the placenta and accumulate in fetal tissues, but research is limited on how they may affect offspring cardiovascular health. Of the few studies that did examine the association of in utero exposure to these metals and elements with child blood pressure, the findings were mixed, and most studies used the one metal at a time approach and did not take into account metal co-exposures. So why do we need to focus on metal co-exposures? This is because metals and trace elements may have synergistic or antagonistic effects on health. And actually, studying the health impacts of exposure to multiple agents on human health has been listed as a priority in the latest National Institute of Environmental Health Sciences (NIEHS) strategic plan. But it is so far unclear how in utero co-exposure to metal mixtures may affect child blood pressure. So the aim of this analysis is to assess the impact of in utero co-exposure to heavy metals, lead, cadmium, mercury, and trace elements, manganese and selenium on child blood pressure from 3 to 15 years. We used data from the Boston birth cohort, one of the largest and longest running birth cohorts in the United States, comprising a predominantly urban, racially diverse, and low-income population from Boston, Massachusetts. Our study population was 1,194 mother-child pairs. Our exposures were heavy metals and trace elements measured in maternal red blood cells collected 24 to 72 hours after delivery. Our outcome was child blood pressure measured at the most recent annual well-child visit, which fell between 3 to 15 years of age. We calculated child age, sex, and height specific blood pressure percentile based on the 2017 American Academy of Pediatrics hypertension guideline. We used linear regression models to examine the association of exposure to each metal with child blood pressure, and the Bayesian kernel machine regression method to examine the effects of metal co-exposure on child blood pressure. So the BKMR is a novel approach to study metal mixtures in which child blood pressure will be regressed on a flexible function of metal mixture components. And this allows us to examine the potential interactions of the metals and the nonlinear effects of the metal mixture on child blood pressure. Here are some key maternal and child characteristics of our study population. We can see that 61% of the mothers were non-Hispanic Black, 20% were Hispanic, and 53% of the mothers were overweight or obese prior to pregnancy. And for children, 24% were born preterm, 23% low birth weight, and the median age at blood pressure measurement was 8.4 years. So this figure shows the exposure response function for each metal and element with child systolic blood pressure percentile, with all the other metals fixed at their 50th percentile, estimated by the BKMR models. We can see that there is essentially no association of lead, mercury, or cadmium with child systolic blood pressure. but there is an inverse association of selenium and manganese with child systolic blood pressure. And the findings were consistent when we compared the results from the BKMR models versus the results from the linear regression models. In the multivariable adjusted linear regression models, per doubling of maternal selenium and manganese are respectively associated with a 6.23 and 2.62 percentile lower systolic blood pressure. And we adjusted for those covariates listed on the slides. We observed an interaction between manganese and cadmium in association with child systolic blood pressure. We can see from the left panel that the inverse association between manganese and child systolic blood pressure was stronger at higher cadmium levels in a dose-dependent fashion. And similarly, we can see from the right panel that there is a positive association of cadmium with child systolic blood pressure when manganese levels are low. So given that cigarette smoking is a common major source of cadmium exposure, we examined if maternal smoking during pregnancy modified the association of manganese and child systolic blood pressure. And we did find an effect modification here. So on the left panel is a figure from the BKMR model. The red line shows the mothers who smoked during pregnancy, and the blue line shows the mothers who did not smoke during pregnancy. So it was estimated in the multivariable adjusted linear regression model that among mothers who smoked during pregnancy, per doubling of maternal manganese concentrations were associated with a 10.09 percentile lower child systolic blood pressure. But among mothers who did not smoke during pregnancy, there was no association of manganese with child systolic blood pressure. So our findings suggested that optimizing in utero selenium levels, as well as manganese levels in women who had higher cadmium or who smoked during pregnancy may protect offspring from developing high blood pressure in childhood. One of our major strengths of the study is that we used the Bayesian kernel machine regression method to assess the nonlinear effects and the interactions of metals and elements on child blood pressure while accounting for metal co-exposures. And had we not used those methods, we may have missed the important interaction between cadmium and manganese. So this emphasizes the need for future studies to focus on how metal and trace element co-exposures affect health. I would like to acknowledge all collaborators and funding sources of the study, and thanks for listening. I'm happy to take any questions at the live Q&A session afterwards.